the, okay, and you can hear me, right?
Yes.
Okay, so good morning to everyone in India and whoever is in this, whoever is in the Indian Standard Time Zone. And I believe that there are people who are from other time zones also. So for them, good evening and good afternoon. Before I start my talk, I would like to thank the group of Bioingene for creating such an amazing platform. When Shubhabrata contacted me to present my work, I asked him about the target audience, and he sends me this pie chart. And actually, I was really thrilled to see the pie chart. I felt privileged, but at the same time, it was a little bit nerve-wracking because you can see the diversity in the audience. And then I thought, what should I present to this audience? Should I present my postdoctoral work or should I present my PhD work? And while thinking about it, it was during the time when the lockdown was taking place in US, and I was at home, and I thought probably if I talk about plant-virus interaction, it will be more relevant in this situation. So I selected this topic on plant-virus interaction. So in today's talk, what I'm going to talk about is, first I am going to show why is plant-virus interaction important, what is plant-virus interaction. Then I will tell you a short story on plant-virus interaction. This is basically a small part of my research work that I did during my PhD. And finally, I will discuss how can we apply the knowledge of plant-virus interaction. Now, as I noticed that the target audience is diverse, so my first two topics are just very basic backgrounds on plant-virus interaction. So let's start with why is plant-virus interaction important? Now we all know that the human population is increasing rapidly. And with the increasing rate of the increasing population, there is a need for more food. And the question remains is that how can we address this increasing demand of more food? To me, and I think most of you also will think that there might be two strategies to solve this problem. First, by increasing the yield of crops. And we are hearing about different talks in this webinar series about how to increase the yield of crops, and there will be more that will talk about this. The other way is by reduce crop losses. And this is where plant-virus interaction comes into play. Now, why plant-virus interaction? Now, plant viruses are known to cause diseases worldwide to agriculturally important plants. And with the change in the climate, the geographic range is continuously increasing. It is estimated that every year, plant virus diseases cause crop losses up to $30 billion. And still we find that viruses are difficult to control. So we need to make sure, or we need to continue research on plant viruses and their host, and understand the interactions so that we can develop novel disease control strategies. Now, in line with this, just last week, when I was preparing this talk, I saw this particular paper that was published in Molecular Plant. So it's a comment paper, and it talks about, the title of the paper is Pandemics of People and Plant, which is the greater threat to food security. I would encourage you all to read this paper because there are some interesting views about plant-virus interaction. It talks about the current COVID-19 situation, but also it brings about the plant-virus interaction. So kindly read this paper. And you will also find more information about the significance of plant-virus interaction in this paper. Moving forward, let us talk about what is plant-virus interaction. Now, what I have done in this section is to to divide it into three subtopics. At first, I will talk about the overview of plant-virus interaction. Then I will talk about one of the important components of plant-virus interaction, that are plant viruses. And I will focus my talk on RNA viruses and their multiplication cycle. Finally, I will talk about the outcomes of plant-virus interaction. So how can we define what is plant-virus interaction? To me, plant-virus interaction is the study of all the mechanisms that underlies the outcomes of these interactions. For example, if a virus infects a plant and the plant develops disease. Now, if you are studying how this disease developed, what are the mechanisms that are taking place, what are the harmful effects, then this is what we study in plant-virus interaction. The other part of plant-virus interaction is also to understand how environment plays an important role in determining these outcomes. So this is mostly, in a simplified term, the plant-virus interaction. Now I will be talking about viruses. Now, what are viruses? Plant viruses are ultra-microscopic agents. They multiply only within living host cells, and also for their multiplication, they hijack the plant cellular machineries. The basic components of the viruses are proteins and nucleic acid, and the viral genome can be either RNA or DNA. So here on the right, you can see electron micrograph of a particular virus called tomato ring spot virus. And these are spherical viruses. And in the bottom, I have a cartoon of the spherical viruses where you can see the viral genome that is encapsulated by a viral code protein covering called capsid. I know all of you remember this, but I thought probably before I get into the details about multiplication cycle of a RNA virus, let us refresh a little bit about the central dogma of molecular biology. So we all know that DNA gets transcribed to RNA, and then RNA gets translated to protein. The virus today I will be talking about, like the multiplication cycle or in the later parts of my talk, is a positive sense single-stranded RNA virus. So what I mean by this is that the viral genome is a RNA. And so from here, the viral RNA can get directly translated into proteins. Let's get a little bit better view of the plant RNA virus multiplication cycle. Now, when a virus infects a plant, the first thing that it does is it releases its viral RNA. This viral RNA is then translated into proteins. Some of these proteins are then used to make multiple copies of the viral RNA. Once the multiple copies are formed, at the same time, some of this viral RNA, in association with some of the proteins called viral code protein, they both assemble to form new virus particles. So this is how basically the virus is multiplying itself. Once it forms these particles in association with other accessory proteins, the virus moves to neighboring cells. And eventually it moves to the different parts of the plant. So this is how within one particular plant, the virus infects first few cells, and then it basically spreads all throughout the plant. The next question then gets raised is that how does the virus infect a new healthy plant? Now, viruses need vectors to get transmitted to new healthy plants. And these vectors can be insects, nematodes, or even us human beings. So this is basically a simplified view of the multiplication cycle of RNA virus. But we have to remember a couple of things. So in the previous slide, I showed you how the virus multiplies. But we also have to remember that in a cell, there are other cellular processes taking place. And that is what the virus has to take into consideration. For example, what the virus has to develop different strategies to hijack the plant machineries. That is something it must do, because why will the plant suddenly let you hijack its own plant machinery and then let the virus multiply, right? Also, in addition to this, there are plant defense responses that are present within this plant cells, and the virus has to escape or it has to inhibit this plant defense responses. So you can see that the plant virus multiplication cycle actually is much more complicated than I showed you in the previous slide. And the interaction between these different processes actually determines the outcomes of the plant virus interaction. Now, so what are the outcomes of plant virus interaction? To me, broadly, there can be three different outcomes. The first outcome is a case where the plant is severely damaged. Its growth is damaged. It might be damaged to an extent where the plant completely dies. And this is a type of interaction that really worries us. Suppose if you have an economically important plant and that develops a disease where the plant dies, and that's how you get huge crop losses. For example, this is an example, this is an example of a virus infecting cherries. So in the oval, in this red oval, you can see cherries that are not infected by virus, but then you see this other cherries that are infected by virus, and the size of the cherries are affected. And this is affected by a virus called little cherry virus. So you can understand why this virus was called little cherry virus. This is another example where we can see potatoes being infected by potato virus Y. Suppose you go to the market and you see this particular potato. Will you pick this up and bring it to home? And then are you going to eat it? So you can see you will try to avoid this kind of potatoes. Again, you are affecting, again, these viruses are affecting the economy. Another example that I really like is the viral infection caused by cassava mosaic virus on cassava. Now, cassava is currently the third most source of calories in the tropic. And the basically what we are interested in eating from the cassava is the tuber. So you can see here this tuber, usually that's what we human beings eat. But then you can see here in this oval circle, this red circle, that hardly has any, that has a stunted tuber caused by a virus infection. And this again causes huge economic losses, and also this can reduce the yield of the crops to a huge extent. So this is our first kind of outcome where the plants are highly damaged. The second kind of outcome basically is the effect where there is hardly any effect on the plants, where the virus is restricted or it is completely cleared away. This type of outcome can be called resistance. And this mostly takes place in plants where they have particular kind of a specific kind of gene called resistance gene that is specific, that specifically can block the multiplication of a particular kind of virus and restrict its growth within the first few inoculated cells. A classic example of this is the interaction between the tobacco mosaic virus and the N gene. And I would like to tell you all a story because I really like this story. So this was in the year 1930s in New Jersey. Um, what people were finding, well, not people, the farmers were finding is that they were growing tomatoes, and the tomatoes were developing these symptoms. Like here you can see this yellow colored symptoms. Also on the fruit, they were getting this mosaics. This was later found to be caused by a virus called tobacco mosaic virus. And the question was that from where the tobacco mosaic virus is getting infected into these plants. Because in New Jersey, there were no tobacco plants, but they were not cultivating tobacco. Any guesses? If this was in person, I would ask you all. If you have any ideas, you can write it in the chat box too. The answer was this particular cigarette called Double Happiness. What was observed was that the farmers working in the field will go out, they would smoke the cigarettes, and these cigarettes were actually infected by the tobacco mosaic virus. And they would get back into the field and they will work, and unknowingly they will be infecting the tomato plants, and that was causing the disease. This is a picture of a tobacco plant that was that's infected with this particular virus called the tobacco mosaic virus. And let me tell you that tobacco mosaic viruses are the first are the first discovered viruses, and that is how the field of virology, I would say, developed. And why is this? You can see that because tobacco mosaic viruses are very stable. It has been reported that they can be stable in the environment for like 50, 60 years. So that is one of the reasons why this virus was one of the viruses that was discovered at the very beginning. But coming back to our story of tobacco, so here you can see that the tobacco mosaic virus is causing great symptoms on this particular tobacco plant. But you won't see the symptoms on all cultivars of tobacco. In certain cases, in certain cultivars, the virus is restricted to a few cells only, like here as shown in this particular lesions. And the reason behind is because these are plants that have a resistance gene called the N gene that basically blocks the virus or restricts the virus within this first few inoculated cells. This other picture is a top view of the same plant, and you can clearly see that all the other leaves are basically symptomless. So the virus is restricted only in the first few inoculated cells. So this example is obviously a great example. Later on, this N gene was introgressed into tomatoes, and there are transgenic tomatoes that express this N gene, and they are resistant to the tobacco mosaic virus. So far, what I have talked about is about the outcomes is that on one extreme, you have a case where the plant survives, the virus is cleared. So you're getting resistance. On the other end, you have the plant dies, the virus multiplies, and this is called susceptible. But there are more, but this is like the two extremes. But more and more examples are emerging where viruses and plants, they are found to be existing in between these two extremes. And this is called tolerance. In tolerance, what is interesting is that the viruses are trying to cause less damage to the plants. And in the same way, plants are helping, or not helping, but the plants are allowing the virus to remain in their cells. And one thing I should say is that somehow it is being noted, it is to be noted is that they are providing advantage to each other. Let me give you an example. This is an example of an interaction between white clover and white clover mosaic virus. Now, white clover mosaic virus shown here in the middle are the white clover mosaic viruses. They infect white clover plants. Uh, it was interesting to find that there are this other insects called fungus gnats. Now, fungus gnats are small fruit fly-like insects. They lay their eggs on organic material in the soil, and these eggs in moist conditions hatch, and then then the larvae that comes out from these eggs, they start eating the leaves of the plant. So eventually the larvae can eat the whole, all the leaves of the plants, and eventually can kill the plant. So this is really a huge problem. But interestingly, what was observed is that the fungus gnats, the larvae of the fungus gnats, were somehow not preferring plants that were infected with this particular virus. So the virus was helping the plants to get rid, or somehow was creating a condition where the insects didn't like these plants. One of the reasons that was suggested is that these plants, they release certain kind of volatile compounds. And once you have this in the virus infected plants, what was observed is that in these plants, the volatile compounds that are released, that they are released in a different amount or different profile, and somehow the fungus gnats were detecting this and they were avoiding these plants. So here, the white clover mosaic virus was to some extent providing an advantage to the white clover plant. Another example that we like to think falls in this category is the phenomenon of symptom recovery. So what is symptom recovery? And symptom recovery is one kind of plant virus interaction. In this case, what you find is that the infected cells, they develop symptoms, followed by symptoms on the upper uninoculated or uninfected cells. Eventually, new leaves emerge, and these new leaves are recovered from the infection. And they don't have symptoms. That's why we call them recovered from the infection. They are symptomless. And this phenomenon is called symptom recovery. In this case, we like to add it because we like to add it to the tolerance outcome, because these plants can bear seeds. And I don't think that there are reports showing that there's a difference in the amount of seeds that being produced. So there's less damage on these plants. Also, the other reason why I'm discussing this here is because this is what I worked as a PhD student. So this was my PhD topic, and this is what I studied. And that's what I'm going to talk about in the next few slides. And I'm going to tell you, while studying symptom recovery, how we were able to discover a novel antiviral defense response system in plants. So now we all know what symptom recovery is. This phenomenon of symptom recovery has been observed in several plant virus interactions. It was first reported in the year in the early 20th century. But the mechanism underlying symptom recovery was discovered only in the late 1990s. Dr. David Balcomb's lab in Cambridge, they were studying the interaction between tobacco blackring virus and Nicotiana clevelandii plants. They observed that the recovered leaves contained lower levels of viral RNA. So what they found is that the viral RNA is getting cleared in the recovered leaves. They also found out the mechanism that was involved. So what they noticed was that in tobacco blackring virus infected plants, the RNA silencing mechanism, which I am going to get into the details in the next few slides, cleaves the viral RNA. And finally, the viral RNA is cleared, and that causes recovery. So recovery was attributed to viral RNA clearance by RNA silencing. So let's get into a little bit of details about RNA silencing or RNAi. So what is RNA silencing? Now, RNA silencing is a molecular mechanism to regulate the expression of genes by small RNAs. This is present in diverse organisms like fungi, plants, animals, and insects. Now, RNA silencing can take place, or it can have its effect, on different parts of gene expression. If RNA silencing acts at the transcriptional level, it is called transcriptional gene silencing. If it acts at the post-transcriptional level, it is called post-transcriptional gene silencing. For today's talk, I will focus on post-transcriptional gene silencing, and I will be referring to it as RNA silencing. So let's get a little bit more about the process of post-transcriptional gene silencing, and now I'm going to call it as RNA silencing. So how does this process work?